Hello everyone. I'm going to talk about some more science today, if that's all right. So I'm just going to do that. Now, the just see here now. Yes, I want to just talk about sort of bacteria and viruses and things like that today. And I just want to write this out for you very quickly, if you don't mind. Just want to show you this. That's a disinfectant, antiseptic disinfectant, and that just ties in with what I want to talk to you today about. So yes, I want to just right. Barbicide, disinfecting, killing of most microbial life, disinfectant solution used by barbers for disinfecting tools. So I just thought I'd mention that to you. Germicide, a substance or other agent which destroys harmful microorganisms. So that's a germicide. And fungicide, a chemical that destroys fungus and viruses. So I just thought I'd say that to you. Disinfectants, to kill harmful bacteria and viruses. So I just thought I'd say that to you. Disinfectant is effective for the disinfection of viruses and bacteria. So I just thought I'd say that to you. Right, so yeah, barbicide is a disinfectant, I think, and a fungicide is used to kill parasitic fungi, and a germicide, a substance or other agent which destroys harmful microorganisms. So I just wanted to say that to you. These things are what I'm talking about. Are capable of destroying harmful microorganisms. So a barbicide is a disinfectant solution used by barbers for sanitizing tools. So I just wanted to say that to you. And that what I've written down there is something about destroying harmful bacteria and viruses. I just wanted to say that to you. So if I just so what I shall just a minute. So if I just sort of talk about similar things now, pathogens and disease. Microorganisms are organisms that are too small to see without magnification. There are millions of species of microorganisms, some of which live on or in our bodies. A few are pathogens because they cause infectious diseases when they are passed from an infected person to someone who is not infected. Many pathogens are bacteria, such as those that cause cholera, food poisoning, dysentery, TB, tuberculosis. Other diseases are caused by viruses, e.g. influenza, mumps, measles. A few pathogens are fungi, such as the athlete's foot fungus. And so I just thought I'd meant to say that to you. So and then you have antiseptics and antibiotics. So let's just say we can reduce infection by preventing the pathogen that causes it from getting into our body. using antiseptics, we can reduce infection by preventing the pathogen that causes it from getting into the body. Chemical substances that kill microorganisms outside the body are called antiseptics. Antiseptics can help prevent pathogens getting into an open wound. So I just thought I'd mention that. So yes, I just wanted to say that. So I just thought I'd say that to you. Thank you for that. I'm going to just write another chemical down now. Just want to write this down actually. Thank you for being patient. If I put this down, right, I just want to put that down, write that down for you. That it's another antiseptic or something like that. What I'm talking today about is, I'm talking at the moment about antiseptics, disinfectants, stuff like that. So I just wanted to show you that. Thank you for waiting there. Thank you for that. Right. I'm going to talk about Alexander Fleming today and penicillin. Before 1934, there were few effective drugs. Some herbal preparations may have been useful. After all, many of our present-day drugs are derived from or based on plant products. Quinine, for example, was used for the treatment of malaria and was extracted from a specific kind of tree bark. In 1935, a group of chemicals called sulfonylamides were found to be effective against some bacterial diseases, such as blood poisoning, pneumonia, and septic wounds. Alexander Fleming had discovered penicillin in 1928, 7 years before the use of sulfonylamides, but he had been unable to purify it and test it on humans. Fleming was a bacteriologist working at St Mary's Hospital in London. In 1928, he was studying different strains of Staphylococcus bacteria. He had made some cultures on agar plates, AGAR. And left them on the laboratory bench during a 4-week holiday. When he returned, he noticed that one of the plates had been contaminated by a mould fungus, and that round the margins of the mould there was a clear zone with no bacteria growing. Fleming reasoned that a substance had diffused out of the mould colony and killed the bacteria. The mould was identified as Penicillium notatum, so that's Penicillium. And then it's notatum, and the supposed antibacterial chemical was called penicillin. So that's right, the mould was identified as Penicillium notatum, and the supposed antibacterial chemical was called penicillin. Fleming went on to culture the penicillium on a liquid meat broth medium and showed that the broth contained penicillin, which suppressed the growth of a wide range of bacteria. Two research assistants at St Mary's tried to obtain a pure sample of penicillin, free from all the other substances in the broth. Although they succeeded, the procedure was cumbersome and the product was unstable. By this time, Fleming seemed to have lost interest and to assume that penicillin would be too difficult to extract and too unstable to be of medical value. In 1939, Howard Florey, a pathologist, and Boris Chain, a biochemist working at Oxford, succeeded in preparing reasonably pure penicillin and making it stable. Techniques of extraction had improved dramatically in 10 years, and in particular, freeze drying enabled a stable water-soluble powder form of penicillin to be produced. So World War II was an urgent incentive for the production of penicillin in large quantities. And this undoubtedly saved many lives that would otherwise have been lost as a result of infected wounds. Once Boris Chain had worked out the molecular structure of penicillin, it became possible to modify it chemically and produce other forms of penicillin that attacked a different range of bacteria or had different properties. So that's right. So for example, so this word AMPICILLIN. Now this is another antibiotic. For example, this word is a modified penicillin which can be taken by mouth rather than by injection. Because penicillin was the product of a mould, chemists searched for other moulds, particularly those present in the soil, which might produce antibiotics. So, that's right. The ideal drug is one which kills or suppresses the growth of harmful cells, such as bacteria or cancer cells, without damaging the body cells. That's what I wanted to talk about, and antibiotics today. So that's right. And carbolic acid, which is phenol, PHENOL, killed the pathogens, but was a very strong antiseptic. So it killed the pathogens, but it was a very strong antiseptic, unpleasant to inhale, and it interfered with the natural healing process. So Joseph Lister, who was another pioneer in the 19th century, in the 1860s, 1870s, used milder antiseptics and sterilised his instruments and dressings by heating them, and dressings by heating them. And obviously, Joseph Lister pioneered antiseptic surgery. That's right. So I just wanted to say that to you. Agar, AGAR, a clear jelly extracted from one kind of seaweed. On its own, it will not support the growth of the bacteria or fungi, but will do so if food substances are dissolved in it. A jar with different kinds of food dissolved in it is used to grow different kinds of microorganisms. So I just wanted to say that to you. Bacteria. Right. After the war, Fleming returned to his research at St Mary's. In 1928, he noticed that the Staphylococcus bacteria he had been growing in his laboratory had become contaminated with mould. This mould formed a bacteria-free zone around itself. Fleming soon realised chemicals inside the mould could destroy bacteria. This antibacterial substance, named penicillin, was the world's first antibiotic. So if I just reiterate or repeat it, in 1928, he noticed that the Staphylococcus bacteria he had been growing in his lab had become contaminated with mould. This mould formed a bacteria-free zone around itself. And Fleming soon realised chemicals inside the mould could destroy bacteria. And the antibacterial substance, this antibacterial substance, named penicillin, was the world's first antibiotic. So I just wanted to say that to you. In the 1860s, British surgeon Joseph Lister was the first person to promote the use of hygiene sprays, antiseptics and sterilising equipment to stop the spread of germs. So that's right. So in the 1860s, British surgeon Joseph Lister was the first person to promote the use of hygiene sprays, antiseptics and sterilising equipment to stop the spread of germs. And then, of course, English scientist Edward Jenner performed the world's first vaccination in 1796. Smallpox was England's deadliest disease before the vaccine was introduced. And a pathogen is any organism that causes disease. Diseases caused by pathogens are called infectious diseases. Pathogenic microorganisms include some bacteria, some fungi, and all viruses. So I just thought I'd mention that. Thank you for listening to me there. Just get some. Right. So if I just talk about Alexander Fleming again, right. Yeah. The agar was inside what is called a petri dish, right? So the agar, agar, was inside what is called a petri dish. So yes, was establishing the bacterial agar culture system and the association of bacteria with disease. See, if the bacteria were present on the swab, they would be transferred to the surface of the agar, agar. Then the bacteria would feed on the nutrients within the agar, and would grow atop the agar. The identical procedure used today to isolate unidentified bacteria. These bacteria could later be isolated and studied further. See, as the story goes, one day in 1928, Fleming left the lid off the top of one of his petri dishes for a little too long. Right, so yeah, as the story goes, one day in 1928, Fleming left the lid off the top of one of his petri dishes for a little too long, and a fungal spore landed on it. The culture became contaminated. After returning from vacation, Fleming noticed that his Staphylococcus culture was contaminated with this fungus. But instead of throwing the petri dish away, he carefully examined it first. Fleming noticed that there was a zone around the fungus which was completely devoid of bacteria, whereas elsewhere on the agar, the bacteria were growing just fine. If you pretend that's fine. So yeah, if you pretend that the little white discs are fungal growth and ignore the label, Fleming's petri dish might have looked something like Fleming correctly reasoned that this growing fungus might be producing some sort of diffusible substance which killed the bacteria. He then identified the fungus as Penicillium notatum, isolated the material which killed the bacteria, and named the substance penicillin. Although finding that this substance killed many different kinds of bacteria, and should be highly useful to treat infections, he also found that the substance appeared very unstable. Many times would not work, so he quit doing any research on the substance and published his last work on penicillin around 1931. Time passes, it's around 1935 or so, and still no major breakthroughs with respect to treatment of bacterial diseases, except for the discovery of a human-made substance, sulfanilamide. Now, nobody went out and said, okay then. Right, so yeah, right, so yeah, Heatley, this is another scientist, a biochemist, designed all the experimentation for isolation, purification, and characterization of penicillin. And after, right, that's fine. So, right, that's what I just wanted to say to you. Yeah. If I just say. Just bear with me. Yeah, so stories of people becoming ill, obviously, with streptococcal and staphylococcus, which I'm going to write these down, and the way that this penicillin cured some people. Right. So yeah, so basically, Fleming couldn't extract enough of it to save lives. So, there was a procedure then that found a way to use deep fermentation techniques, which produced 10 times the yield. And I think this was in America somewhere. And pharmaceutical companies began producing penicillin. So yeah, so the to extract more of it, to produce more penicillin, I think they had to use deep fermentation techniques, which produced 10 times the yield of the people back at Oxford. I think the famous university. So yeah, that's right. So just wanted to say that to you. Yeah, that's right. So basically, during this time, the original mould discovered by Fleming was being used for the production of penicillin. A new type of penicillin mould was then found on a mouldy melon from a market somewhere in America. This mould gave more penicillin than Fleming's mould, which meant that penicillin production could be stepped up in time for the D-Day landings. And that there was enough penicillin to treat all the wounded British, American and other Allied troops. So I just wanted to, so there was now enough penicillin to treat all the wounded British, American and Allied and other Allied troops. So I just wanted to say that to you. Thank you for listening to that. Yeah, so if I just talk about again how penicillin is discovered. He said so. Basically, Alexander Fleming sort of revolutionised all medicine by discovering the world's first antibiotic or bacteria killer. So this penicillin, which is an antibiotic, was the first antibiotic or bacterial killer. So yeah, he was a Scottish. Alexander Fleming was a Scottish microbiologist, and he left when he absent-mindedly he was going on holiday, and he absent-mindedly left an amount of Staphylococcus bacteria on a tray in his lab. On his return, he noticed that a patch of mould had stopped the bacteria's spread. He realised that a substance in the mould, which Fleming called penicillin, had antibiotic qualities that could stem the spread of chronic infections. The number of lives subsequently saved are countless. So I just wanted to say that to you. Right, thank you for that. Right, so if I just... Right, but bacteria comes comes in three basic shapes: spiral, spherical, and rod shaped. So if I just write this down for you. Right, yeah, thank you for your patience. Yeah, three basic shapes: spiral, spherical, and rod shaped, which is bacillus. I think rod is bacillus, or bacilli, and it's like sort of like sausage sort of shaped bacteria. Yeah, under obviously a very powerful microscope. You've got spherical, which is round, and you've got spiral. So you've got three: there's spiral, spherical, and bacillus. And rod is actually bacillus. And then you've got two famous ones, which obviously the one that Fleming was working on was the spherical ones, spherical bacteria. And I just want to write that down for you, actually. So if I just show you that, have I shown you that? I don't know, thanks. Right, I just wanted to write that down for you. So now, if I just put this down. I think this was the one that Alexander Fleming was working on, the Staphylococcus one. And as I mentioned, I think that's spherical. These ones are spherical bacteria. So yeah, I just wanted to say that to you. And I just wanted to talk about something else, something to do with that now. So yeah. Now you've got gram-positive and gram-negative bacteria, and I think the Streptococcus is gram-positive. I think it is, and and Staphylococcus is also gram-positive, I think. Yeah, it is. Just wanted to say that to you. And yeah, so just wanted to say that to you. And salmonella is rod shaped, I think. So it's like the sausage shaped bacillus one, and it's gram negative, I think. Salmonella is gram negative, I think, and typhoid is gram negative, and it's rod shaped. Is it? I'm not sure actually. I'm not sure. Salmonella is rod shaped. I don't know about typhoid, but it's gram negative. And these bacteria basically thrive in where there's poor sanitation, contaminated water, so probably that got contaminated with human waste and feces and stuff like that. Undercooked food as well. So it's basically common sense, but obviously the pioneers of the 19th century had to sort of prove it to the lay people that they were right, and some of the not just the lay, some of the qualified people didn't believe these pioneers of the 19th century, late 19th century, early 20th century. So it was a tough time for them. So yes, and Clostridium, I think, is gram positive bacteria. So Listeria is gram positive. It's rod shaped again. And obviously, so Listeria is gram positive. It's rod shaped, and it's it's bacterium caused by eating food contaminated with bacteria, obviously. And I just want to talk about now. Another thing I want to say now is that most bacteria are classified into two broad categories: gram positive and gram negative. These categories are based on their cell wall composition and reaction to the gram stain test. The difference between gram positive versus gram negative bacteria are primarily related to their cell wall composition. Gram positive bacteria have cell walls composed mostly of a substance unique to bacteria known as peptidoglycan. Right, I'm just going to put this. Sorry, here. I'm just going to write this down for you. Right, so it's their cell walls here, and the word is that it's plant cell walls are cellulose, but the bacteria ones aren't cellulose. This is the word I'm going to write down for you. I just want to write that down for you. Yeah. So that's what the cell walls are made of. Peptidoglycan or murine. These bacteria cell walls, they're not, so it's in contrast to the cellulose cell walls of the plant cells. So I just thought I'd say that to you. These bacteria stain purple. Right, so Gram-negative bacteria have cell walls with only a thin layer of this word peptide, the one I've already written down, and an outer membrane. So yeah, the gram-negative bacteria have cell walls with only a thin layer of the peptide thing, peptidoglycan. And gram-positive bacteria have cell walls composed mostly of a substance unique to bacteria known as peptidoglycan or murine. So yeah, it's. Just wanted to say that to you. Yeah, it's. You might have to just consult the textbooks or a bit more with the difference between gram negative and gram positive. So I just wanted to talk about that. Right, thank you for. Right, and just another thing I want to say. Name some ways in which microbes can enter the body. Well, respiratory, digestive, and broken skin. Just wanted to say that to you. So yeah, I just wanted to say that to you. Thank you for listening to me there. If I just. And another thing I just want to say before I finish with you today. So yeah, if we talk about diseases which probably can be cured now, perhaps not a few years ago they weren't, but they're eradicated. They're cured now. Some of these diseases I want to talk to you about. So polio, I think that's a virus derives from a virus. Measles derives from a virus. Chickenpox derives from a virus. Tetanus derives from bacteria. TB, tuberculosis, comes from bacteria. Food poisoning is bacteria, and pneumonia is bacteria, I think. And antibiotics works with bacteria, but it's not effective with viruses. So I just wanted to say that to you. So, obviously typhoid, cholera, dysentery, diphtheria, tetanus, are just basically derived from, I think those ones derive from bacteria. So I just wanted to say that, and I think that's, just want to say a few more things before I wrap things up with you today. So I just want to write some things down, and then I'll finish with you, if that's all right. So just wanted to say, right, I just want to write one thing down, and then I'll finish with you, if that's all right. Thank you for being patient. just got some antibiotics down here, a list of antibiotics. Some of the early ones, actually, that were discovered. Is it discovered? I think 1930s, so these are ones that were discovered in the 30s or early 40s. I just wanted to show that to you. Thank you for your patience today, and I hope I've explained some things to you that you perhaps didn't know. That's very nice of you. And if you like my channel, could you subscribe to my channel? It'd be very kind of you if you do. That's very, very nice of you. So thank you very much. Thank you. Bye.